Hi everyone. I would like to talk about X soup or why p-value can be sometimes more than 5%. My name is Alexander Kell, and let's go directly into the topic. So basically, you probably know this very nice cartoon story about button soup, where the girl actually started to make a soup out of one button. But of course, all the important ingredients were then playing important role. So similar story we had about egg soup. It's a much simpler soup where you have fewer ingredients with an important part, of course, the eggs, which is here, but you have also onion, potato, bay leaves, salt. But let's consider each ingredient separately and estimate how eatable is actually each individual ingredient and estimate it in a form of p-value. Let's say onion. I like onion and I eat it sometimes as it is. And let's put a p-value of eatability, like can I eat this? About 0.001. And potato, fresh potato you can hardly eat, but sometimes you can. And let's put p-value about 5%, a bit lower. What about bay leaves? Well, they not really eatable. You can eat it though if you if you like, and we put it above 5%. And salt with a spoon, no. Let's put it 0.2, and X itself is not eatable for sure. But now all these ingredients together, they make a soup. So, and without any of these ingredients, except the last one, maybe your soup is not really good. Like you cannot put, you can create, make your soup without salt, but salt is indeed a very important ingredient, although individually by itself, it's not really eatable. So the same is with our studies of very complex gene regulatory networks. When we analyze individual genes in our gene expression data and we compare disease versus control, and we would estimate a p-value of each individual gene to be important for that particular disease or that particular condition. But important to say, although like on this picture, the whole function, the whole network is much more complex. And if individual genes, individual proteins on this diagram, like those ones which are in red here, they are significant by themselves. But some, like this superoxide dismutase 1, SOD1, appear to be very important for the whole function of that network. But when we estimate the p-value of that individual protein, it was above 5%. So that's the hero of our story today. But let's go to the story. Basically, the study was done around the famous compound called 5-FU, fluorouracil, which is a very important and well-known drug, which is used to treat many different cancers. But what is important to know and to remember that this drug, when it's applied, it develops the cancer develops actually resistance to this drug. And for instance, for colorectal cancer, about half of the patients, they actually, metastatic colorectal cancer, about half of the patients, they actually develop the resistance to that drug. So that's why for us it was important to understand the mechanism of resistance. And together with my colleague in Stockholm and Karolinska Institute, Roman Zubarev, we actually did several experiments. He mainly did it in his lab. He took several cancer cell lines, and the HCT 116 is actually colorectal cancer cell line. And by treating this cell line with iteratively with increasing concentration of the 5-FU, he developed a resistant cell line. So those cell lines which are actually resistant to quite high concentration of 5FU. They still live and still proliferate. So, and then we were interested to understand basically what is the difference in the mechanism of cancer cell line growth in the resistant cell line versus sensitive cell line. And if we understand this mechanism, perhaps we can actually find a second compound, a second drug, which we can combine with 5FU to overcome this resistance. And this is a data which he produces. He actually have a very advanced proteomics facility, which can measure expression proteomics, standard expression quantitative proteomics, and they measure more than 7,000 proteins in that experiment in these sensitive cell lines, making different biological replicates, and also in the resistant cell lines. And there's another method called PISA, or it actually evaluates not just expression of the proteins, but more like an activity of proteins. So these two measurements, they were important for our experiment. And there you have it done for resistant cell lines and for sensitive cell lines. What we did next is actually connected to a very philosophical concept which was developed by Indians many thousand years ago, and that's called prapancha, which is actually, if you translate it to English, is called conceptual proliferation. And essentially, they found out that in our consciousness, many things or many thoughts can be proliferated by itself. So they kind of self-enhanced. And they realized that that's property of our consciousness to be self-enhanced is actually a basis of many of our sufferings, if not all. What we found out and what we saw that actually this concept of increasing self-proliferation is very general, not only for consciousness, but it's actually an important feature of many complex systems in the world. And actually, when those systems got into the bad conditions, into some catastrophe or in chaos, they actually, because of their structure, and the main reason of that, self-proliferation are presence of multiple positive feedback loops when the things which they appear in the system, some disturbances in the system, they can proliferate and increase, and that leads to a lot of catastrophes or the chaotic behavior of the systems. And the more complex the systems, and the more positive feedback loops it has, the more probability to get in such attractors. We believe that this happens also with biological systems, with cells, and with the whole organisms, and that's one of the basic, actually, underlying principle of many of our diseases, especially cancer. When the tumor cells they start to self-initiate, self-activate, enhance, and which leads to uncontrolled proliferation. And this also happens, we believe, in the difference between resistant and sensitive cells towards a drug, anti-cancer drug. So basically, the resistant cells they found a way to proliferate under condition of treatment of that drug. So that's they rewire the whole system towards a different situation, but still they found basically a lot of positive feedback loops which maintain this state. All right, so going back to the molecular mechanisms, what we found to be important is to understand how the whole system of gene expression control is organized in the cells, especially in tumor cells. And if we would understand this general mechanism, we would probably find a way how to interact with it, how to trigger the difference which we need. So, for instance, we can measure gene expression in the cells using transcriptomics, or for instance this expression proteomics, which is done by my friend Roman Zubarev. So which can, by comparing one state like say, sensitive state of the cells versus the resistant, we can find out upregulated or downregulated genes or proteins. So those which actually change their expression behavior in two different conditions. The question is why? So we know that gene expression changes are controlled by transcription factors, which are binding to the places in regulatory regions of genomes, in promoters, in other regions. And where do they bind? So we can find using epigenomics perhaps, and we can also find it by applying our algorithms. I will talk about this a bit later. Once we understand which transcription factors are important for changing the expression of our genes, then we can go upstream and ask a question why they're changing the activity, those transcription factors, why they became active suddenly in that new conditions. And by tracing, the signal transduction network upstream from transcription factors, we can find, we call them master regulators, few signaling molecules, few regulators which can control a lot of other changes in the cell. And proteomics, such as PISA proteomics, which actually checks not only presence or absence of the protein, but also testing its activity. It helps also to navigate through the signal transduction network. Finally, those master regulators, we believe, are very good targets for intervention, because if you target such a master regulator, you can influence a lot of processes in the cell. All right, so how do we actually find those transcription factors which are regulating expression of the genes? That's the whole story. Just to make it short, so we have an algorithm which finds clusters of binding sites in promoters of those genes, and we think those clusters they behave like a anchor point for a complex of transcription factors which was not there when the gene was silent, let's say in the condition of sensitive cell line, and then while the cells are evolving towards becoming resistant, those genes they start acting, which means maybe some repressors which were repressing expression of those genes are getting away, but those transcription factors which get active, which are activated, they start finding their target place in the promoters. So which means those target places, they have to be there. So then once they found that, they bind them together in a complex and they activate the gene. So when we find those transcription factors which were working together in binding to those target places, we then, as I said, searching for master regulators, and then once we found them, we can actually search for the new drugs which can interact with those master regulators. All right. So, and again, to explain this concept of feedback loops, on this example, we can say that in the sensitive cell line, there are some feedback loops, maybe positive, maybe negative, which are controlling this state. And many of these positive feedback loops, they allow these cells to proliferate. But while this transition happens to resistant state, the whole network is now rewired to find the new actually feedbacks, new positive feedback loops, which allows these cells to proliferate under the condition of that treatment. And now we can try to find those master regulators. So which means they not only have to be able to lead to the activation of the transcription factors, which we found important in those regulated genes, but we need to also to confirm that those transcription, those regulators, those master regulators, are under the direct control of the genes which they actually activate. So the positive feedback loops. All right, so having such a condition and putting this all in our pipeline, in our algorithm, first we wanted to check whether this algorithm actually works. And we took data from cancer cell line encyclopedia, which if you know, it's a big repository of various omics data, especially we can find RNA-seq data and genomics data for multiple cell lines, like 500 cell lines, cancer cell lines. And we have also data about the sensitivity of those cell lines to various drugs. And in this experiment, we choose one drug, which is lapatinib, which is a drug which is targeting EGF receptor, and we choose list of sensitive cell lines which are sensitive to lapatinib, and list of resistant cell lines, and we took RNA-seq data and compared the resistant cell lines to the sensitive cell. Actually, sensitive cell lines towards versus resistant. We wanted to know those genes which are upregulated in sensitive cell lines. So, and then we run this search for transcription factor clusters in the promoters of those sensitive of the genes which are upregulated in sensitive cell lines. And then we run this master regulator analysis upstream from the transcription factors, and we found master regulators, and EGFR was among the top master regulators found by the system. And respectively, when our system started to prioritize the drugs which are from our collection of drugs and their targets, then this lapatinib was actually the top drug in our prioritization. So we actually confirmed on this positive example that the algorithm works. So now we wanted to apply to this new data, so where we actually don't know the mechanism. So now we are comparing resistant cells towards sensitive. So sensitive, they are sensitive to 5-FU. So we know 5-FU kills them, but resistant, they are not really well sensitive to 5-FU. We need to find new drug targets in the resistant cell lines. And we applied the same algorithm. So we found, with expression proteomics, those genes. Actually, we found proteins, but we also think that expression of the proteins mainly controlled by the increased expression of the genes, in such a setup. And then we found those genes. And then we found with PISA, with activity proteomics, we found those proteins which get active in the system. And then analyzing all this multi-omics data, we came up with a network, and we found master regulators. So this network was quite big, and here you see just the top of that. So the top candidates. And as I explained already before, basically we found that those proteins which are shown here in pink with a red hour around them, they are upregulated, and the p-value of upregulation is very well significant. But one of the master regulator, which was one of the most important master regulator in the whole regulatory cascades here, superoxide dismutase 1, SOD1, actually, the p-value of this individual gene was not very significant, not significant at all. It's 0.28. So if you would actually through this gene from the very beginning, we would not find it in this analysis. But actually, we found out that it's a very crucial part, because when then we applied our drug prioritization, and our first focus was on those actually compounds, not really drugs, but chemical compounds which would not be very toxic, because as you know, if you combine various toxic drugs, then toxicity would alleviate, and it will be unbearable. So then the idea would be to have like such a toxic drug like 5-FU combined with some compound which would be completely untoxic. And among those very quite untoxic things, we found Uracil is one of our top candidate. And this SOD1 found to be a target for this Uracil. So basically, Uracil is just a chemical structure. You see it here; it's pretty small, but still, it can bind to various proteins. And with using chemoinformatic tools like PAS, using structure activity relationship, database, which is behind this PAS system, we predicted that Uracil can be a compound targeting this SOD1. And not only, so there are several drug targets found among our master regulators, which are a target for Uracil. So Uracil was our one top candidate. So, and indeed, we know the SOD1 is important for cancer biology. So it's one of the key enzymes which actually reduce reactive oxygen species. And by the way, another enzyme, which is this GPX, was also on our actually diagram. This is glutathione peroxidase 1, which by itself actually was significantly changed. Expression of that protein was significantly changed, and you see in this line, in this chain of reactions, that's just the second enzyme after SOD1. So, and of course, to remove reactive oxygen species and to convert them to water, both reactions are important, and the second reaction is driven by this enzyme, which actually change elevated expression in resistant cell line. So basically, in resistance, to increase resistance of the cell line, we see that one of the mechanisms which cell develops is to accelerate them, accelerate the to removal of reactive oxygen species. And we can see that by inhibiting SOD1 with some compounds like Uracil, we can cut this. Metabolic chain, and we also can change this whole regulatory signaling events as well to reduce this all kind of feedback loops. All right, so and actually, as you can see here, we show this enzyme as well. But in fact, when this Uracil combination with 5-FU was quite well known, it was in early application of 5-FU, people already found out that by combining it with Uracil, they get a better, efficacy. And but then this Uracil was substituted by a real new drug, which was developed on the basis of Uracil, which was much more effective. And actually, this new drug was even higher in hierarchy, in ranking than Uracil itself, although it has higher toxicity, perhaps. Well, at least in our computations, it has a higher toxicity, predicted toxicity. So, but it ranked higher, so which supports again that our predictions are really true. What is interesting, actually, and that's indeed shows again that this molecule, which was developed by GSK, is indeed enhanced the therapeutic effect of 5-FU. All right. So coming back to SOD1, we found this paper which actually shows that there is a interesting post-translational modification which SOD1 can have in the tumor cell line, in tumor cells, which is accelerated by acetylase SIRT1. So it actually SOD1 in active form after SIRT1 is acetylating it. It actually brings it to inactive form, and that's actually happens in the sensitive cell lines And as you can see, in the, here we see this SIRT1, actually on the border of my slide, which is downregulated in resistant cell lines. So which means, SIRT1, by bringing SOD1 into inactive form, it makes it more sensitive. That's why here in the resistant cell lines, the SIRT1 is downregulated. So the cell line gets it downregulated. Again, it confirms that SOD1, although it doesn't change expression, and as I said, if we would simply, be based on p-value, and we would skip this gene at all because it doesn't change expression, but the change is happening on the post-translational level. So when this protein is actually modified by another protein, and this way is actually, resistance is maintained. And okay, so Uracil is here predicted as one of the top candidates, and with our collaboration with Roman, he did actually mix the Uracil with 5-FU on that resistant cell lines, and validated and shown in the experiment indeed this combination works better, and the sensitivity to 5-FU is resumed, and 5-FU start killing those cells effectively upon treatment by additional Uracil. Finalizing my lecture, I would say that indeed such tools like I presented, which applies some bits of AI, I would say more like machine learning, are really helping us to fight cancer. And my message here again: so when you prepare your soup, don't forget to salt it. So otherwise, it's not digestive. The same here. Don't throw away your genes just because the p-value is below threshold. Find another, confirmation which will show you that all these proteins can work together in one system and help each other. All right, with that, I would finish, and thank you for attention, and see you next time.
By the way, people ask, what about X? What kind of role it plays in this analogy? And I say the X is actually 5-FU. We started with 5-FU to develop a resistant cell line, and we analyzed it, and we discovered a mechanism which actually works for resistance under various conditions. So basically, the X is not needed.